Hello, everyone, and welcome to our October virtual series. I'm Sandra Franco, and I am the Education and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. So as you know, in this virtual series, we highlight new papers and methods that stem from our center and that we think that might be of interest for the broader community. Today, we have two speakers that have worked together to repurpose and validate a piece of equipment that most of you will be familiar with, the HiSeq2500 instrument, and turn it into a versatile fluidics and imaging platform. As usual, there's going to be time at the end of their presentation for you to ask questions, and you can do so by either raising your hand or typing your question on the chat box. With that, I'll introduce our pair of speakers today, Kunal Pandit and Joana Petrescu. Kunal is a research engineer at the Technology Innovation Lab here at the New York Genome Center. He obtained his PhD at the University of Maryland, where he worked under the supervision of Srini Raghavan and Ian White on hydrogels, microfluidics, lab-on-chip devices, and biosensors. At his current position, Kunal is now focused on the development of methods for spatial multi-omics. Joana is an MD PhD student at Columbia University and a member of Hemalis Phatnani's lab at the New York Genome Center. Her project seeks to understand changes in cell state and intercellular interactions that are reflected by pathological features such as TDP43 aggregation in ALS frontotemporal dementia spectrum disorders. With that, I'll hand it to the both of you.
Thanks, Sandra. So I'm Kunal. Like Sandra said, I'm a research engineer in the Tech Innovation Lab, and I'm going to talk about a subtitle of our talk could be Teaching a HiSeq2500 New Tricks. So first off, I'd like to acknowledge Will Stevenson, who originally was in our lab and has now moved into Genentech. And he originally had the idea to hack into HiSeq and play around with the instrumentation inside. And I'd also like to acknowledge Hacteria, and which is this open source bio group mostly based in Europe. And they have a project called the Resig Lab, where they want to develop an open source sequencer and hack HiSeq2000s and 2500s to do sequencing in an open and cheaper manner. So the HiSeq2500 was released a few years ago, and it was the workhorse of production labs across the world. But now it's been obsolesced by newer sequencers. And Illumina has even announced they will stop supporting the instrument and supplying the reagents after early next year. So now they've been decommissioned by numerous places, including here at the Genome Center. We have a bunch of them upstairs that are just collecting dust and are now even available on eBay to buy. So instead of just letting them collect dust and not doing anything with them, we thought it'd be cool to see what's inside and see if we can make it a useful instrument again. So at its core, the sequencer is just an epifluorescent microscope with integrated fluidics, which means it really has the potential to be a versatile and valuable piece of scientific equipment. Inside, there are lasers, filters, cameras, total control for the stage in 3 directions, and valves and pumps, and everything's integrated together in one robust machine. So we wrote software called HiSeq2500 to control each of a lot of the different modules inside and develop an automated control system where you input an instruction file, a config file, and it will go through the instruction file, configure the instrument in whatever way you'd like, run the experiment, and output images and logs so you can fully automate an experiment and walk away. So I'll just go through some of the modules that we wrote control software for and automated. The first is the stage module. So it has a stage with slots for 2 different flow cells. You can move the stage in the X and Y direction with very fine resolution. It also has 3 tilt motors that move the stage in large Z movements up and down. And also you can have some rough control of the tilt of the stage. These are these 3 motors at the corners here. And then there's also a piezoelectric actuator connected to the objective for very fine Z resolution. And the objective is a 20x air objective with a 0.75 NA. In the X, Y, and the objectives direction, there's excellent recall that you can really go back to the same spot over and over again, which means, and that and combined with the fact that there are vacuum lines in the stage to hold flow cells in place, which means that you can really, there's not much need for image alignment after imaging multiple cycles. Also on the stage are, like I said, there are the vacuum lines to hold the flow cell in place. There are alignment rods so that the flow cell is aligned properly every time you take it off or take it off and put it back on. And there are fluidic ports for the inlets and outlets, and there are gaskets to create leak-proof seals. And then underneath the stage, there's also a temperature control module. There are peltiers and cooling liquid can flow under the stage to adjust the temperature of the stage. So here we set the stage to various different set points, and we achieve pretty rapid and heating and cooling. And we measure the temperature of the individual flow cells from the HiSeq, and also we use an external thermocouple to read the temperature. And the HiSeq usually runs hotter than what we set it to, depending. And we think that's because to compensate for any offset for the temperature inside an actual flow cell. And the temperature module for each, temperature control for each flow cell slot on the stage can be independently controlled. The fluidics module consists of a bunch of valves and pumps. So buffer is pulled through from reagent tubes through different, through a series of 2 different ports or valves in through the flow cell and then out of the flow cell into the syringe pump. So the buffers can be chilled in the onboard chiller on the HiSeq, and there are 24 ports on the reagent valve. So, but on the stock HiSeq, only ports 1 through 8, 10 through 19, and 20 are actually plumbed. We modified the HiSeqs to add more reagent lines at ports 9 and 21 through 24. We also modified the syringe pump a little bit so that each barrel is normally plumbed to each lane in a HiSeq flow cell. So there's 8 barrels, and each barrel has 250 microliters capacity. But we plumbed the 2 outlets of our flow cell to all of the inlets of the syringe pump so that we have the full pumping volume of the pump to be, so which is now 2 mils. And the fluidic module is replicated for each flow cell and can be independently controlled. And then lastly is the optics module. So there's only one set of optics module for each flow cell, but it comprises a bunch of different things. So there are excitation lasers, a green and red excitation laser, and the intensity can be controlled up to 1 or 2 watts. And you can also control the intensity with excitation filters and neutral density filters. And the laser lines are then directed through the objective, and fluorescence emission is collected and filtered by 4 different bandpass filters at those wavelengths, and then directed to 4 TDI land scanning cameras. And we can acquire simultaneously images from all 4 cameras, and we can control all these different components and synchronize them to do 4-color imaging. So with these excitation lines and emission filters, finding fluorophores is a little bit challenging though. The emission bands are pretty tight, so their spillover is inevitable. We can, for example, we can excite electrophore 532 and electrophore 594 with the green laser, but emission from the 532, there's significant spillover into the adjacent channel with electrophore 594. Similarly, with we can illuminate or excite Psi5 and electrophore 700 with the red laser, but spillover from Psi5 into 700 is a problem. So to deal with this and do simultaneous 4-color imaging, we used an algorithm called Picasso, which I won't go into too much detail, but essentially it minimizes the mutual information between channels that have spillover to remove the spillover and have mutually exclusive sort of stains. And here I'm showing that with mixed signals, and then after unmixing, the magenta comes through much nicer. And this is what we can get. These are the kind of images that we can do with simultaneous 4-color imaging. The resolution is sub-micron resolution in the X and Y direction, and it's really nice pictures. So the cameras are these TDI line scanning cameras. They're a little different than cameras I was used to. So TDI stands for time delay integration. It's a special readout method for CCD cameras where the object is moved under the sensor, and as the object passes each sensor, the exposure is integrated for each line that it passes. And so it's a way of integrating the exposure for fast imaging and high sensitivity. And we can really do some pretty fast imaging with this field of view and this kind of camera. So the field of view is 768 microns by 6 microns. So that's basically the length of the line, the width of the line, but we can acquire arbitrary long tiles just by moving the object in arbitrary long distance. So we can do stacks of tiles that are pretty big. And the images that are output are 12-bit TIFFs. We also wrote an autofocus module for true walk-away automation. And the way that works is it first takes an autofocus image at a pre-programmed Z position, and then it looks in the autofocus image for field of views with the most contrast and filters out the field of views with the most contrast. And then we rank those field of views by distance away from that are distance from each other. And we go through those field views and find focal positions until a consensus focal position is reached for the section, and then image the section at that position, or a stack we're doing a stack, a series of images above and below that position. So what I mean by finding the focal position, and we take an objective stack where this is sort of a stretched out view of the field of view from just like one line. And we continuously image the section as we move the objective away from the section. And then each of these frames we convert into a JPEG. And when you convert it into a JPEG, the larger the file size of the JPEG image, the more in focus the image is. And so this is a trick that I picked up from NASA, which they use on the Mars Curiosity rovers and other rovers for quick autofocus finding. So you can see on the right here, at the bottom of the stack, there's not much signal and it's all over the place, but as it gets more in focus, you can see some nice airy discs. And then at the middle position, at the objective step 28930, there's just really pinpoint beads, which is what we're imaging here. And then as you move away from the focal plane again, they get blurry, and then finally there's no signal. And on the left is just the JPEG size as a function, as we moving the frame number, moving the objective away from the stage. So we really aim to use a stock HiSeq with minimal modifications so other people could easily use this software. And the only modification we had to do was remove this vanity plate that has these little notches that I've circled in red that don't allow you to use regular glass microscope slides. So you can remove that plate by just removing 3 different screws, and that's the only mandatory modifications to get our homebrew flow cells onto the machine. The other modifications that we did were, as I said, we added more lines to the reagent valve, so we had more reagents to play with, and we drilled a hole on the side of the HiSeq and routed the lines to the side. And then we also added manifolds to the pumps to take full, so we're able to pump the full volume of all 8 barrels. To do automated control, like I said earlier, you have to just specify a config file and a recipe. And in the config file, you outline what experiment you want to run. You measure your sections that you want to image, and I'll show that in the next slide how we measure regions of interest to image. And then you specify all the reagents and which port they correspond to. And if you want to do cyclic imaging, or cyclic reagent exchange, where every cycle you bump in a new different imager or reagent, you can specify that too, and specify filters for every cycle of imaging, and all sorts of different settings, like the flow settings, how you want to do the focusing and whatnot too. And all this is documented in our GitHub for people to really tailor their own experiments to. And so this is how we measure the ROIs, and this is an example of our flow cell. So on the left is a HiSeq flow cell with 8 lanes. In the middle is our flow cell design without any tissue section onto it, and we use 2 inlets and 2 outlets, and again we tie all the outlets from the HiSeq into those 2 outlets on our flow cell, so we have full volume of the pumps. And then when we section tissue on, we print out a slide ruler or grid onto a piece of transparency. And then we just measure out a lower left and an upper right bounding box of the section and input that into our config file. And the HiSeq will go back and calculate where the stage positioning for each region of interest and image the entire section. So just to wrap up, we wrote CICA2500 to automate imaging and fluid exchange of flow cell experiments, and we try to write it in a way that is generalizable to any experiment. Currently, we're assisting others to get started, for example, over at Cold Spring Harbor and at the Broad to do 4i and other methods. And I'm adding new features every week. Mainly right now, we're adding just more diagnostic features to troubleshoot any problems with the HiSeq. And we're also adapting new protocols to put in the HiSeq besides 4i, which Joanna will now talk about.
Yeah, so my name is Joana. Thanks, Kunal. I'm a graduate student in the Phatnani group here at the New York Genome Center and also at Columbia University. And I'm going to talk a little bit more about how we use iterative indirect immunofluorescence imaging to really show off all of the things that this CICA2500 code base that Kunal developed can do. So first, 4i was developed by the Pelkman's Lab, and it allows you to visualize dozens of proteins in a single sample using unmodified off-the-shelf antibodies. And as you can see in this image, the Pelkman's group developed this method for use in cultured cells, but we wanted to adapt it for highly multiplexed immunofluorescence of mouse and postmortem tissue sections. We thought this was a particularly appealing method because it would allow us to customize our own antibody panels using not only cell type markers, but also antibodies for pathological features that had been clinically validated. And so just to give some more background on the 4i method, it really was based in this very clever observation that was made by the Pelkman's group. And so to walk you through this image, essentially what they did is they conducted a typical immunofluorescence experiment, and then they imaged just a region of interest that's outlined here in this orange box. They then used a protein denaturing buffer to remove and elute the antibody from the sample, and then added back the same secondary antibody in order to see where the primary antibody they had used remained. And so really what they saw when they imaged this sample after this elution and restaining with the secondary antibody was that they were able to remove the primary antibody everywhere except the ROI that had been imaged. And so this led them to hypothesize that during, you're using these very bright lasers during confocal imaging, perhaps this causes reactive oxygen species, which can then cause cross-linking of the antibody to the sample. And so they basically developed this 4i chemistry, and the chemistry is centered on trying to prevent this photocross-linking of the antibody with the epitope. And so to walk you through the changes that are different between 4i and a typical immunofluorescence experiment, the first is that you conduct imaging in an imaging buffer, and this buffer acts as a reactive oxygen species scavenger and also as acceptor for this photocross-linking reaction instead of the primary antibody. This allows you to use a really mild elution buffer to denature the antibodies without degrading the sample that you're staining. And then lastly, they add malamide to their blocking buffer to block the sulfhydryl groups that they found to be particularly prone to cross-linking. And so with this approach, you're basically able to take one sample, conduct an immunofluorescence experiment, image it, elute the antibodies easily from the sample, and then keep doing this iteratively in order to build a highly multiplexed view of where proteins are in a sample. And so in our lab, I worked to adapt this method to work on intact mouse tissue sections, which you can see here, where we stained for multiple cell type markers across 3 cycles of 4i. And you can see already that just, a few cycles of 4i, and we overlay some of these stains, we already start to see a really detailed view of the tissue composition of the sample. And so this preliminary experiment gave us confidence that we can apply 4i to tissue sections. But we kind of ran into an issue, which is that 4i essentially entails multiple sequential immunofluorescence experiments, each of which lasts around 8 hours plus imaging time. And so we were really looking for some sort of open source, adaptable approach to automating this protocol. And I was very lucky that I worked closely physically at the New York Genome Center, very close to Kunal and Will and everybody else in the tech innovation group who thought that we could adapt a HiSeq to do these experiments. And so the really cool tool that they have developed enables us to basically conduct automated unattended 4i on multiple samples in parallel, thereby increasing our throughput. It allows us to stain the samples repeatedly. And so these buffer exchange and imaging steps are automated so that they are always taking the same amount of time. And then lastly, we conduct real-time logging to ensure experimental reproducibility and accurate sort of sharing of our experimental protocols. And so as I mentioned previously, the real central advancement of 4i is that you conduct imaging in this reactive oxygen species scavenging buffer in order to prevent photocross-linking of antibodies to the sample. And so this really, if you're doing this in cultured cells, you can do this in a tissue culture plate that has, a bottom that is compatible with microscopy. But if you want to do this on intact tissue sections, this sort of presents the challenge of how do you exchange buffer over the sample in a way that is compatible with imaging. And so our first challenge was really that we wanted to build a flow cell in order to allow us to automate this method. And so of course this flow cell had to be compatible with a HiSeq because we wanted to use the HiSeq and the CICA2500 code base in order to do this method. But we also really wanted to make sure that we were using low cost, really accessible method or components to make the flow cell, and that it was sort of adaptable to other applications. And so briefly, the way that we sort of make our flow cells is we start with a standard microscope slide that has been custom drilled with holes that align with a CICA2500 stage like Kunal already showed. We coat these slides with polylysine to facilitate tissue adhesion. Then we cut a flow cell spacer that creates the chamber out of this double-sided polyamide tape. And this spacer is about 100 microns thick, which allows us to fill the chamber and flow liquid over tissue sections while also maintaining the proper working distance between the objective and the sample. And so once we get to this stage, we can section our tissues into the flow cell and store the whole thing at -80 until we're ready to use it. After we fix the sample and we process the tissues, we can then create a closed chamber by removing the adhesive backing from this flow cell and adhering a cover glass that then creates the full assembled flow cell that we use on the HiSeq. And so, as I already mentioned, we really think that this is an adaptable design. If you change the coating or the shape that you cut out of this Kapton tape, we think that it could be applied in a number of different ways. And so when we were optimizing the 4i protocol to be performed using HiSeq2500, we first had to determine the optimal number of incubations with the elution buffer in order to fully remove antibodies from the sample. And so you can see here, this is our staining with GFAP and eLav-L2. And after just the first incubation with the 4i elution buffer, we see a very quick drop off in the signal close to the background signal. So for all of our subsequent experiments, we felt confident that 4 incubations with the elution buffer that are 10 minutes each is sufficient to fully remove all of the antibodies from the sample. But we also wanted to make sure that once you remove these antibodies, the sample remains intact and can be restained. And so we basically tried to replicate one of the experiments in the original 4i paper on tissue. And so what we did is over multiple 4i cycles, we alternated between even and odd cycles between different primary antibodies, but in every single cycle we used the same secondary antibody to make sure that we had fully removed the primary antibody from the previous round. And so as you can see here, after each cycle we can fully remove the antibodies from the previous round and yet restain the same tissue section with a new antibody, and we can do this iteratively over multiple cycles of 4i. And so as I just talked about, we have this flow cell design, but because it is modeled or based on a standard microscope slide, it's quite large. And so this allows us to process multiple tissue sections in parallel. And so we wanted to demonstrate that you could use this to your advantage in experimental design by sectioning transgenic and control samples onto a single flow cell and processing them at once in order to minimize technical variability. And so here you can see some spinal cord sections from a non-transgenic mouse, as well as a SOD1 G93A ALS mouse model. And they were stained with a number of different cell type markers over 4i cycles. And this was done all on one flow cell in order to minimize the contribution of experimental variability to the difference in the staining that you observe. But then we wanted to take it one step further. So not only did we process non-transgenic and mutant mice on the same flow cell, we actually also sectioned adjacent 10 micron sections on 3 different flow cells that we then processed both on the 2 different places on the stage, like flow cell stage positions, but also in 2 different experimental runs. So yeah, so as you can see here, for multiple stains, the variability of the staining within a genotype across both flow cells and experimental runs is less than the variability between the non-transgenic and the mutant animal. And so we really think that the HiSeq2500 automation will facilitate comparing between biological groups by reducing this technical variability. And then lastly, we show that HiSeq2500 enables automated unattended 4i on postmortem spinal cord sections from patients with ALS. Importantly, the HiSeq2500 setup allows us to conduct multi-day experiments on multiple samples in parallel and to image these really large tissue areas that are almost, a centimeter in each dimension with subcellular resolution. And so just to sort of recap this whole presentation, we used 4i to really demonstrate the power of HiSeq2500 automation in order to make a complex multi-day protocol that involves both fluidics and imaging be really high throughput and reproducible. So in addition to increasing the throughput by allowing multiple samples to be processed in parallel and showing that we've gotten repeatable buffer exchange and imaging between flow cells and between experiments, we also show that you can conduct simultaneous 4-color imaging of these very large tissue sections with micron resolution. And then lastly, we're really proud that this is done with an open source code base and a flow cell design that we think is adaptable to other fluidics and imaging based methods. And as Kunal mentioned, we're already working to make sure that other groups can implement both 4i and other methods on the HiSeq system in their own labs. So with that, I again want to thank everyone at CGND, especially Miguel Cuevas, who collected all of the spinal cords from mice that were used in this study. And then of course, Kunal Pandit, Will, and everyone at the Tech Innovation Group who helped develop the HiSeq system. And that's it from me.
Thank you. Thank you so much, Kunal and Joana, for sharing your work with us. And now we're going to move to the Q&A section. So if you have any question, you can either raise your hand or you can type your question on the chat box. I have tons of questions and, and I think I'm going to get started. So the first thing I'm curious about is, can you use, so I know, Joana, you mentioned that one of the ideas to repurpose this was to perform 4i in tissue, but can this instrument also be used to stain culture cells? I don't know.
Yeah, that's a great question. We haven't done it yet, but one of the groups that we're working with is interested in doing this, and we have a number of thoughts about how that could be done by either growing the cells directly on the cover glass like you would, or maybe even seeding them into the flow cell that we have designed, but we haven't directly tested it yet.
Yeah, we're like about to try that next. Actually, we're about to try to grow cells on our flow cell. And see if we can fix them to the flow cell and see if they stick after flowing buffer through the flow cell.
Yeah, and my other question is about the ROI. So you have to input at least where your sample is. Is it possible also maybe to just do your imaging on a certain regions of your tissue? Like, for example, if you would be interested in, I don't know, ventral horn or dorsal horn just, would it be possible for you to just restrict the ROIs that you want to image instead of imaging the whole sample and then maybe speed up the process?
You could just measure that ROI yourself. We haven't built any sort of like on-the-fly adaptive imaging except for the autofocus. But the imaging time, especially if you're just doing one Z-plane, is fairly fast. We can probably image a whole human section in under 10 minutes with one Z-plane. So
yeah, and I think especially with my tissue sections, like you said, I think it would be very easy to manually measure the ventral horn because it's visible by eye. I think if you were to try to select an ROI on the basis of like the actual microscopy image, that's something that, as Kunal's saying, we would need some sort of adaptive tool to be able to do that. But if it's a feature that you can pick out by eye, I think it's pretty easy to zero in.
Yeah.
Nice. And then the other thing I was wondering is, do you see, because in the protocols, like I read that the imaging protocol can take up to 6 days or something like that, like 4 or 5 days. Do you see like a decrease in the quality of the tissue, like on the quality itself? And would you recommend maybe people to start cycles with those antibodies that have worse signal and then end up with those that have better signal?
Yeah,
definitely. Yes.
Yeah, that's great. Yes, to all of this. That's a great question. Yeah, we definitely, especially because we're using fresh frozen tissue and postmortem tissue that is fresh frozen is also very fragile. We definitely see that after 6-8 rounds, we see some degradation in the tissue itself. And so we kept it below that number, but also I do think there's some variability in the stability of the epitopes that you would have to optimize for new antibodies and make sure that if you're using it in a later round, the signal is the same as if you were to use it in the first round.
Yeah, that makes totally sense. And I guess this is the last question. Where do you see this technology moving forward? Like what other applications one could use or kind of repurpose your protocol? Yeah.
So we're adapting it for spatial multi-omics. We want to do proteome, transcriptome, and maybe some genotyping in tissues and in cells. We're specifically adapting SABR, immunoSABR and SABR RNA and DNA, because it's the same chemistry to measure all three modalities. We're also interested maybe in doing optical pool CRISPR screens where we read the CRISPR edit in situ and have measure a few antibodies too, maybe to get a morphology of the cell in conjunction with a single cell experiment too. But yeah, we're thinking about like multi-omic spatial omics methods and also kind of use combined modalities with single cell.
That's awesome. So thank you again, Kunal and Joana, for sharing your work with us. It's really exciting work, and also as you highlighted, it's open source, so anyone potentially could adapt it for their work. So that's really, that's really nice that you share that with us.